Hi, this is Aaron, and welcome to Aaron Explains. So today I'll be talking about my favorite biological process, photosynthesis. Specifically, I'll go over the first half of photosynthesis, the light reaction. As I go through, I'll put up some big picture ideas in the upper right corner in red there. And the first of these is that photosynthesis is energy conversion. Photosynthesis happens to turn energy from the sun in the form of photons into usable chemical energy later on. So, how do plants even do this weird process that turns the sun into sugar? So the first problem they have to overcome is capturing the energy of photons, and plants do this by using pigments, which are molecules that absorb photons. So, when a photon is absorbed by one of these pigment molecules, it excites electrons inside of the pigment molecules, and it's these high energy electrons that can be used to do work. Plants have several different types of pigments. The most common of these, which makes plants green, are called chlorophyll. And there's two types that are very similar, chlorophyll A and B. And there's also another group called the carotenoids, which kind of gives them a bit of an orangish hue, although they're a lot less common than chlorophyll. So where do these things actually fit into the plant cell? So I want to bring up, or draw rather, a little blob thing. And it's not going to look very pretty right now, but don't worry, it gets better. This is the chloroplast. It's the plant-only organelle where our grand process happens. And inside the chloroplast, we have these stacks, or grana, made of folds of membrane called the thylakoid membrane. And it's this thylakoid membrane where all of the action of the light reaction is going to take place. So I'm drawing a little close-up of some stylized thylakoid membrane. And I'm going to talk about some of the major players as they come along. And we're going to be moving from left to right, just for reference. So the first thing that you're going to encounter on your journey through the thylakoid membrane is photosystem 2. This is probably going to need a bit of explaining. So, this is a photosystem. It's a cluster of proteins with different functions. You can think of the structure kind of as a jelly donut. There's an outside and a middle part. The outside is made of the light harvesting complex. And these little green dots that are inside the light harvesting complex, which are wonderfully, wonderfully depicted, are chlorophyll molecules. I know, I'm the next Van Gogh, right? Now, the inside is also really important to our little photosynthesis journey. And this inside part is called the reaction center. Finally, something that's named cool. It contains a special chlorophyll A molecule, which is basically two chlorophyll A molecules bonded together, and a region called the primary acceptor right above that. I know this seems like a tangent, but bear with me, this is important. So after photosystem 2, which by the way is also called P680, based off of the nanometers of light that it best absorbs, we have this region called the ETC. The ETC is an abbreviation for electron transport chain, and its name is fairly descriptive of what it does. It just transports electrons from one side of it to the other. And I'm not going to talk too much about what this part does, other than that it acts as a shuttle, but I will mention that the large central molecule there is called the cytochrome complex, and I'm just going to abbreviate that as CC. And I'm going to redraw the cytochrome complex in red, just so that we have a bit of continuity as we go on through. So after the ETC is photosystem 1, which is like photosystem 2, except that it absorbs 700 nanometers of light better. And we have another very short electron transport chain, but this one literally does nothing but transport electrons. And after that, we have this little blue box, and this is the NADP plus reductase molecule. So I'm just going to mark this off in blue too. And now we have our little cast of players which are going to act out the process of photosynthesis. So let's go and try to expand on this smaller picture, and I'm going to make it a lot bigger so that we can go and add in a lot more details to fully describe what's going on here. So just a quick overview again. We have photosystem 2, followed by an electron transport chain, followed by photosystem 1, try to keep that one in mind, a second electron transport chain, and NADP plus reductase. Awesome, so let's get this show on the road. I mentioned before how chlorophyll makes electrons excited, so the first order of business is to obtain electrons. This is where that wonderful liquid water comes in. So it turns out that photosystems can actually hydrolyze, or split water. So in comes water, and out pops two hydrogen ions, an oxygen atom, and two electrons that are drawn into that special chlorophyll A molecule that I mentioned. That oxygen that comes out pairs with other lonely oxygens, and together they form oxygen gas and they leave the cell. And that is what makes plants, algae, and cyanobacteria the great oxygenators of the planet, and the reason why we're all alive. And these protons have another role to play, so I'll get back to that. So the chlorophyll molecules are excited by light, and they start to play this game of energy hot potato, you could kind of say, where the excited amount of energy from one chlorophyll molecule will then jump to another one that's nearby, and then keep jumping in between these chlorophyll molecules until it reaches that special chlorophyll A molecule. And once there, it excites those electrons that we just got from water, and they jump to the primary acceptor. Now, these electrons are now chock full of energy, and that energy, unfortunately, is not going to last past the next stage. In the next part of this process, our really energized electrons are going to go into the electron transport chain, and they're going to lose that energy that they just gained. And I'm going to talk more about why they lose the energy when they enter the cytochrome complex in just a minute. But for now, we now have these electrons that are depleted of their energy, and we now have to re-energize them in order for them to actually be of use. So those depleted electrons are then shipped off to the next part of our little thylakoid journey, photosystem 1. And the process here is essentially the same as photosystem 2. A photon of light comes in, it excites chlorophyll molecules, they play hot potato with the energy, and they eventually reach that special chlorophyll A molecule that has our two electrons with it. The electrons are excited, and they go to the primary acceptor region of photosystem 1. And now after that, our excited electrons go to a second, very short ETC, and they don't lose any energy this time. It's just a transport mechanism. They merely pass on through to the next and last part of their journey, at least in the light reaction, NADP plus reductase. So this thing's job is to combine the molecule NADP plus with a proton and the two electrons to make NADPH. So in a way, this new molecule can act as sort of an electron ferry and bring these two energized particles to the next phase of photosynthesis, the dark reaction, or Calvin cycle. So why did those electrons lose energy in the first electron transport chain anyway? It's because they were doing work to pump hydrogen ions into the interior of the thylakoid membrane, against the concentration gradient which exists, with a low H plus concentration outside and a high H plus concentration inside. And the reason why this gradient exists is because the H plus will then flow through this molecule called ATP synthase. And that energy of its flow will then be used to combine inorganic phosphate with ADP molecules to create ATP, which is the general energy unit of cells. And ATP2 will be going on to the Calvin cycle or dark reaction, because photosynthesis is very multifunctional, and I'll write that up there in a minute. But what I just described of this entire process of photosynthesis is called linear flow, because electrons go in one end and they come out the other. So what if this process is backed up? What if the NADP plus isn't made to take away those charged electrons? Well, this situation where there's sort of like a backup of electrons actually happens a lot more than you would expect. But don't worry, there's a very ancient backup plan. And I'll be the first to admit that sounds pretty cool to say. You gotta try it sometime. So, instead of getting rid of those energized electrons and having them taken away by NADP+, now that the NADP+ isn't actually there, those energized electrons are just going to be shipped back to the first ETC to keep pumping hydrogen ions inside and maintaining that proton gradient across the thylakoid membrane. Because as long as that gradient is maintained, you can still make ATP, or usable energy if you want to think of it that way. And the electrons just keep cycling around, getting energized, then pumping, then energized, and you get the idea, just continuously. This process, however, is a lot less efficient than going through the whole photosynthesis to make sugar as an energy source. This is called cyclic flow, and it's used pretty much only in a pinch, and they think this is a very ancient and primordial version of photosynthesis. And that brings me to my last point, is that evolution isn't super efficient, but it is very good at patching up what it once had. So I hope you learned a lot about the first half of photosynthesis today, and I look forward to talking to you guys again soon. Bye everyone!